Good day, my dear grade 10s. Today we're on the topic of the chemistry of life, grade 10 life sciences. Yes. So on the last video, we've covered the topic on the organic compounds and the inorganic compounds. This time we're going to cover the food test using food types. Yes. So such as the testing of carbohydrates, lipids, and proteins. So we're going to start with the test for carbohydrates. So the test for carbohydrates, if you remember, are the one that contains monosaccharides, polysaccharides, and also what we call diaposaccharides. Yes. So first and foremost, when testing for carbohydrates, they're going to be a usage of Benedict solution. Yes. So Benedict solution is a test for glucose, my mistake. Benedict solution is going to be a test for glucose. So each and every time a blue liquid is indicated, it is used to test a glucose. Yes. So as we go down, you can see that here they're indicating the ways of preparing that specific test. You can prepare a water bath and heat the water, pour 5 centimeter squared of Benedict solution in a test tube, a few drops of fruit juice to the Benedict solutions, place the test tube into the water and observe. If the color changes to orange red color, it indicates with a high concentration of glucose inside. So as you can see here, positive Benedict test is going to indicate orange red, and negative Benedict test is going to indicate what blue. So each and every time this solution gives you a orange red solution or color that is present, it will indicate that there is glucose in that, there is a high concentration of glucose in that specific sample. So yes. As we go on, we're going to the test for starch. Yes. Test for starch, it is what iodine solution. So this iodine solution, it is what a brown liquid. It is the one used to test for starch. So if you place a potato or a piece of bread in a dish, obviously we all know that a potato is what has starch. Place a few drops of iodine solution in the peeled potato. The potato will turn blue black. That blue black will indicate what the presence of iodine solution. So the blue black indicates what there is a presence of starch in the specific solution. So yes, each and every time they ask you each and every time they ask you that what are the solutions for starch, you can indicate that it is the iodine solution that is used to test starches. Yes, because this type of questions they love to pop them out in examinations or finals that give us the test for glucose. Then you can immediately say Benedict solution. Test for give us a test for starch. Again, you can immediately say what iodine solution. Then you go into the start for fats, which can be known as lipids. This kind of testing will require you to place a small amount of the test materials in a tube, and you add about 5 centimeters of ethanol. Yes, for the solution that is going to be used for testing fat is going to be ethanol. Check the test tube and let it stand for a few minutes. Filter the test solution and place a few drops of filtrate liquid that filtered through on a clean filter paper. The ether or ethanol evaporate and a transparent fatty stain will form on the paper. If only a solution or if only solution consists of fats or lipids, it will indicate what a stain on that specific filter paper. So the solution that is used to check for fats is what ethanol. So with that, my grade 10s, I would like to conclude this lesson. Hopefully that there is my mistake. I felt that there was something that was missing. So the test for proteins is the test for proteins was the Burette test. So you test proteins by the Burette. So each and every time that may ask you what is the solution used to test for proteins, you can say the Burette. I'm not sure that I'm pronouncing it right, but then bear with me. Grind the test material beans and add a little water. I'll show you the diagram on the side soon. Boil the test material until it forms a thin soup. pour about 5 centimeters of the soup inside the test tube and a few drops of dilute copper sulfate solution to the test tube. Hydroxide solution to the mixture. Shake. This information is not that relevant. It is not needed to be known. The only thing must know that it is the indicators. So each and every time it indicates blue, it will indicate that it is negative. Proteins are absent. Each and every time it indicates purple or violet, indicates positive, meaning that proteins are present. So yes, the only thing that you must know or you must take note of based on this lesson is that the only thing that we want you to know is the indicators. Yes, as long as you know the indicators for proteins, carbohydrates, lipids, and glucose, you're good to go. So as my greetings with that, I would like to conclude this lesson. Make sure that you know your solutions for this test food or this food samples. And I thank you, my greetings. Hopefully you have a great